Welcome to our comprehensive guide on the hit-to-lead process, a crucial stage in drug discovery. This process bridges the gap between identifying promising chemical hits and developing viable lead compounds for further optimization. In this video, we'll walk you through each step, highlighting the scientific strategies and decision points that shape the journey from initial screening to lead selection. Whether you're a student, researcher, or simply curious about how new medicines are developed, this overview will provide a clear and engaging introduction to the world of early drug discovery. Let's start by defining what a hit is in the context of drug discovery. A hit is a chemical compound that shows desired activity in a biological assay, often identified through high-throughput screening. These hits are the initial candidates that could potentially interact with a biological target, such as a protein involved in disease. However, not all hits are suitable for further development. The next steps involve evaluating these hits for their potential to become leads, considering factors like potency, selectivity, and chemical properties. Journey begins with screening large libraries of compounds against a biological target. Automated systems and robotics are often used to test thousands, sometimes millions of molecules rapidly. The goal is to find compounds that show measurable activity, which are then flagged as hits. This high-throughput approach accelerates the discovery process and provides a starting point for further investigation. Once hits are identified, they undergo a series of confirmatory tests to ensure their activity is reproducible and not due to experimental artifacts. After initial identification, hits must be validated to confirm their biological activity. This involves retesting the compounds under various conditions and using different assay formats. Researchers look for consistency in results and rule out false positives. Validation also includes checking the chemical integrity and purity of the hits. Only compounds that consistently demonstrate the desired activity and meet quality standards move forward in the process. This step ensures that resources are focused on the most promising candidates. Once validated, hits are expanded upon by exploring structurally related compounds. This is often done through analog synthesis or by searching chemical databases for similar molecules. The purpose is to understand the structure-activity relationship, or SAR, which helps determine which chemical features are important for biological activity. By testing these analogs, researchers can identify trends and optimize the chemical structure for better potency and selectivity. Structure-activity relationship, or SAR, is a fundamental concept in the hit-lead process. SAR studies involve systematically modifying the chemical structure of hits and observing how these changes affect biological activity. This helps researchers pinpoint which molecular features are critical for activity and which can be altered to improve drug-like properties. SAR analysis guides the design of new analogs and is essential for transforming hits into promising leads. Potency and selectivity are key criteria in evaluating hits. Potency refers to the concentration of compound needed to produce a biological effect, while selectivity measures how specifically the compound interacts with the intended target versus other proteins. Compounds with high potency and selectivity are more likely to become successful leads. Researchers use various assays to measure these parameters, ensuring that only the most effective and specific compounds progress. Beyond potency and selectivity, hits must also possess drug-like properties to be considered as leads. These include solubility, stability, permeability, and metabolic profile. Compounds are tested for their ability to dissolve in biological fluids, remain stable under physiological conditions, cross cell membranes, and avoid rapid breakdown by metabolic enzymes. Hits that fail to meet these criteria are typically deprioritized, while those with favorable profiles move forward. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. Early ADMET screening is performed to predict how a compound will behave in the body. These tests help identify potential safety issues and eliminate compounds with poor pharmacokinetic or toxicological profiles. By addressing ADMET properties early, researchers can focus on hits with the best chance of becoming safe and effective drugs. Chemical optimization is a continuous process throughout hit-to-lead development. Medicinal chemists modify the chemical structure of hits to improve potency, selectivity, and drug-like properties. This iterative process involves designing, synthesizing, and testing new analogs, guided by SAR and ADMET data. The goal is to refine the compound's profile, making it a stronger candidate for lead selection and further development. Promising hits undergo both in vitro and in vivo testing to evaluate their biological effects. In vitro tests use cell cultures or isolated enzymes to measure activity and toxicity, while in vivo studies involve animal models to assess efficacy, pharmacokinetics, and safety in a living organism. These experiments provide critical data for selecting the best candidates to advance as leads. Lead selection is based on a combination of factors, including potency, selectivity, drug-like properties, ADMET profile, and in vivo performance. Compounds that meet predefined criteria are designated as leads and move into the next phase of drug development. This decision is made through careful analysis of all available data, ensuring that only the most promising candidates are chosen for further optimization. Hit-to-lead process is not without challenges. Researchers often encounter issues such as poor solubility, metabolic instability, or off-target effects. Balancing potency with safety and drug-like properties requires careful optimization and sometimes difficult trade-offs. Despite these obstacles, advances in technology and computational methods continue to improve the efficiency and success rate of hit-to-lead campaigns. Computational tools play an increasingly important role in hit-to-lead optimization. Techniques such as molecular modeling, virtual screening, and predictive ADMET modeling help researchers design better compounds and prioritize candidates more efficiently. These tools complement experimental approaches, saving time and resources while increasing the likelihood of success. Summary: the hit-to-lead process is a vital step in drug discovery. transforming initial hits into optimized leads ready for preclinical development. It combines experimental and computational strategies to evaluate, optimize, and select the best candidates. As technology advances, the process becomes more efficient and effective, paving the way for new and innovative therapies. Understanding hit lead is essential for anyone interested in the science behind drug development.